Hello, this is video number 7 for the urinary system. In this video, I will be discussing how the kidneys and the nephrons regulate the pH of your bloodstream. I'll also be talking about a number of other topics, for example, how drugs and hormones, excuse my cat, this is Dinky, are excreted, and some general characteristics of urine. All right, so first topic, control of blood pH. Just a little bit of background first. So blood pH is maintained at around pH 7.4, slightly alkaline. And there are a number of things involved in regulating that. There are buffers in the bloodstream, but the kidney is one of the main ways of regulating and adjusting the pH of the bloodstream. Basically, how it works is if your blood is slightly acidic, the kidney excretes more protons. If it's slightly alkaline, the kidney excretes more bicarbonate. If you recall from the blood chapter in the beginning of the semester, CO2 gets transported in the bloodstream by being enzymatically combined with water to form carbonic acid. Carbonic acid is a weak acid that dissociates into protons and bicarbonate ions. Getting rid of one or the other of these ions will help to adjust the pH and maintain it at the target pH of 7.4. So how is blood pH regulated? It's regulated by special cells located in the wall of the tubule, distal convoluted tubule, the collecting tubules, and the collecting ducts. These cells are called interpolated cells. And basically, they're mixed in with principal cells that I discussed in the previous video. So these cells come in two types. Type A, these cells, what they do is they secrete protons and reabsorb bicarbonate. So the protons being secreted will be lost in the urine, and the bicarbonate will be retained in the body. Type B cells do the opposite. They secrete bicarbonate ions that ends up in the urine, and they reabsorb protons that ends up in the base. Good way to remember these cells and what they do. Type A cells secrete protons, acid, type A, acid. Type B cells secrete bicarbonate ions, base, B for base. Okay, so the blood pH will be adjusted by secreting one or the other of these, getting rid of it from the bloodstream, and bringing the value of the blood pH back to the target range value of pH 7.4. So this slide is just looking at the location of the intercalated cells. It's indicating in the collecting duct here, mixed in with the principal cells, but it's also in the collecting tubule and the distal convoluted tubule. Okay? Now, if we look at the two main types of intercalated cells, so that's shown on this slide. Whoops, sorry. The intercalated cells, this is showing the A cells on the left and the B cells on the right. You can ignore the top part of this slide if you want. It's just illustrating how blood pH can get out of away from the target value of pH 7.4. On the left side, let's consider the situation when the blood pH is too acidic, so less than pH 7.4. When it's too acidic, what happens is the A cells reabsorb bicarbonate into the bloodstream, but they secrete protons into the lumen of the tubule. The secretion is going to end up incorporating, making the urine more acidic, and we're going to excrete more protons while we're saving bicarbonate ions. So that's going to adjust the pH back up. On the other hand, what happens when the blood pH is too alkaline? So greater than pH 7.4. Well, that's where the B cells come into play. The B cells do just the opposite of the A cells. They reabsorb protons into the bloodstream, and they secrete bicarbonate ions into the tubule. Bicarbonate, then we're going to lose more base, and the pH of the urine will be slightly more basic. However, that's going to readjust our blood by decreasing it from the elevated alkaline level. So that's how blood pH is maintained by the nephrons in the kidney physiology. All right, let's talk about another topic. How do nitrogenous wastes get eliminated? First off, what are nitrogenous wastes? So I've talked a little bit about waste molecules. Basically, these are nitrogen-containing breakdown products of either proteins or nucleic acids. And these breakdown products are toxic to cells. So we have to get rid of them. Three main types are shown here: urea from protein breakdown, uric acid from nucleic acid breakdown, and creatinine, which is due to breakdown of a substance in muscles called creatine. Okay, all of these are toxic to cells, and they're constantly being generated, and we must be able to excrete them to get rid of their toxic action. How are they eliminated? Mostly these are dissolved in the urine, and that's the main way that we lose them. For the most part, these molecules are present in the bloodstream. They will be filtered out during glomerular filtration, and they will not be reabsorbed. There are no specific transport proteins in the cells of the wall of the tubule that will bring them back into the body, bring them out of the tubule. Okay, so that is the most common way that they end up in the urine. There is an exception. Urea is somewhat reabsorbed. We're going to ignore that for the purposes of this conversation. How about other substances? So not waste molecules, but other things that end up in urine. So for example, drugs and hormones. So these can be eliminated by one of two ways: either by filtration without reabsorption, so they pass into the filtrate, and they pass the entire way through the tubule and end up in the urine that way. Or by tubular secretion, so they can be added into the tubule by traveling out of the bloodstream and entering the tubule by tubular secretion. So a number of drugs enter into the urine through either of these two pathways. So they're shown on the slide. Antibiotics, for example, various chemotherapy drugs, marijuana compounds, breakdown from absorption of marijuana. Also, some substances such as urobilin. Urobilin is the pigment in urine. It's what gives the urine the kind of yellowish color. That's resulting from the breakdown of the heme group from hemoglobin. That also gets into the urine by, I believe that is through filtration without reabsorption. Another example is the hormone called chorionic gonadotropin, or HCG. This is the hormone that begins circulating during pregnancy, and this is the hormone that's tested for during a urine pregnancy test. This is a protein hormone. So being a protein, it doesn't pass across the filtration membrane during glomerular filtration. The way this protein hormone enters into the urine is through tubular secretion, and then passes into the urine and can be detected by the urine pregnancy test. All right, another topic I want to talk about is diuretics. What is a diuretic, and what are different types of diuretics? So first of all, definition of a diuretic. A diuretic is a substance, a chemical, that enhances the urine output, increases the volume of urine that you produce. So there are a number of different types of diuretics, and these can work in different ways to increase the volume of urine produced. Some examples are caffeine and drugs that are used to treat high blood pressure or hypertension. These work by inhibiting sodium reabsorption. And so as a result, you tend to lose more sodium in the urine. And whenever you lose more of a solute in the urine, you tend to drag along more solvent, water as well. So because of that, there's more volume of urine that you produce. And these have the diuretic effect because of that mechanism of action. Another thing that can cause diuretic, increased urine output, is having a hyperglycemia, so high blood glucose levels. What happens is normally glucose is completely reabsorbed after being filtered. But if you have hyperglycemia, and this could be the result of diabetes, untreated diabetes, the glucose transporters in the wall of the tubule are saturated, and there aren't enough of them in order to remove all of the glucose. So some glucose ends up being in the urine. And just like for sodium, when there's more of a solute in the urine, that drags along with it more water as well. So diuretics, the diuretic effect of hyperglycemia, that's one of the symptoms of hyperglycemia and of diabetes is excess urine production. Okay, another example of a diabetic is alcohol. Drinking alcohol has a diuretic effect. How this works is alcohol is an inhibitor of the hormone ADH. It inhibits the release from the posterior pituitary. ADH, antidiuretic hormone. So if you inhibit an antidiuretic hormone, that causes a diuretic effect. You're not conserving water in the body, which antidiuretic hormone would normally do. By inhibiting antidiuretic hormone, you're losing a lot more water than you normally would. And that's the basis of the effect. The after effects of drinking too much alcohol, you lose a lot of water due to an inhibition of ADH release. And the morning after hangover effect, kind of a dry mouth and things like that. And often headache can be due to dehydration as well. Finally, another type of diuretic is Lasix, and this is inhibiting sodium transporters. So it has a similar effect to caffeine and hypertension drugs. A couple of slides to discuss urine in general, characteristics of urine. So general physical characteristics, fresh urine, so freshly excreted from the body is clear, and but it can vary in the intensity of the yellow color. So if you're dehydrated, you're going to conserve water, and the pigment, the urobilin present is going to be more concentrated, producing a deeper yellow color. If you're fully hydrated, you're going to lose more water, and that's going to dilute the pigment. It's going to be, kind of a less light yellow color. Let's see, some other characteristics. Fresh urine is sterile, but if it sits around for a while, it'll develop kind of an ammonia odor due to growth of bacteria in it. Urine is slightly acidic typically. That can vary somewhat depending on, if your blood pH is being adjusted or not. Here are some things, solutes that are normally found in urine. And mostly this is the list of substances either that are not at all reabsorbed, wastes mostly, or things that are regulated in their absorption. So there are a number of ions: sodium, potassium, calcium also, which is not on this list, ammonia, bicarbonate ions that are normally part of urine. There are also waste molecules: urea, uric acid, and creatinine. Okay, so those are typically substances, solutes that you find in urine. Here's what you don't normally find in urine. And in fact, a urine test is often used to diagnose some disorders that have abnormal substances in them. So normally you don't have any glucose. Glucose is filtered but completely reabsorbed. Presence of glucose, called glycosuria, is an indicator of perhaps diabetes or perhaps excess intake of sugary foods. You don't usually have proteins in urine. Proteins are not even filtered normally. So if you have protein in urine, proteinuria, there can be due to a number of different things. For example, hypertension, high blood pressure can rupture some of the glomerular capillaries that allows proteins to spill into the filtrate. They won't be reabsorbed, so that might result in proteinuria. I'm going to skip over ketones, but hemoglobin is not normally part of the filtrate, right? It's contained normally in red blood cells and they don't pass into the filtrate. Presence of hemoglobin indicates that red blood cells somehow got into the filtrate. So that could be due to, for example, a transfusion, a mismatched blood transfusion is going to cause a lot of rupture of red blood cells and spilling of hemoglobin. And that's one way that hemoglobin can get into the urine. Erythrocytes normally don't get in, but that could be due to bleeding in the urinary tract, for example. Or white blood cells, leukocytes, would be maybe an indicator of a urinary tract infection. All right, I'm going to end this video here. There will be one more video that's going to describe basic overview of the other main structures in the urinary system: the ureters, the bladder, and the urethra, and control of urination or micturition. See ya.